My name is Carol Frost, and I'm an assistant professor here at the University of Alberta in the Department of Renewable Resources. I study arthropod community ecology. Arthropods includes, for my purposes, mostly I'm working on insects and spiders. It includes some other things too, but I focus mostly on insects and spiders. What I research about insects and spiders is questions that are related to their conservation. I believe conservation of insects and spiders, and in particular diversity of those things, which means many species of those things and abundance of those things, many individuals of those animals, is very important. And that's because insects and spiders do so much for us. They contribute a lot to ecosystem function. They do pollination, they do decomposition, predators and parasitic insects attack crop pests, which help us grow our crops, and they do many other important things. They're important parts of the food web that birds and bats eat and fish eat. So insects do so many important things, but we actually know very little about their communities, about how the whole group of insects out there, the whole assemblage of insects, interacts with each other and performs these ecosystem functions. So my research involves asking questions about what insects are out there, what are they doing, and how can we help them to keep doing that? So in a lot of ways, of course, people disturb natural systems. Like we might change what the land is used for through energy development or through forest harvesting or through agriculture. And what I'm very interested in is, of course, we have to do all those things. We have to feed ourselves, we have to produce energy, but how can we do those things in ways that harm biodiversity the least? And my focus is, as I said, insect and spider biodiversity. So questions that I might ask are, like one question that we've asked recently in my lab is, how can we use non-crop land in agricultural areas to best support abundance and biodiversity of beneficial insects, like pollinators and like predators of crop pests? So to answer that question, we've got to go out and find out what insects are in different kinds of field borders. And to do that, we need to, in a standardized way, survey insects. Now, surveying insects, we typically have to collect them, unfortunately. It would be nice if we could just do it like bird watching, where you can just see a bird, identify it, know what species it is. But with insects, they are so small, and sometimes the differences between them are so minute, and you need a microscope to be able to tell species apart. We tend to need to collect insects, bring them back to the lab, and use taxonomic keys and microscopes to identify them. So some of the tools that we use to collect insects are nets. You may not be surprised to find out. So, for example, when we're collecting flying insects, we tend to use, this is called a butterfly net, although I usually use it for bees and flies, anything that's visiting flowers. So we might set a timer, set a transect, which is a set distance of land, an area of land that we're going to walk up and down, and then we walk up and down, watch all the flowers, and anytime an insect lands on a flower, we pop the net over it, catch the insect, and then collect that insect into some kind of a container, label what flower we caught it on, and take it back to the lab. So that's one kind of tool that we use, is just a simple net. You might have one of these at your house. When we're studying insects in aquatic systems, so we have another project studying what kinds of insects are in stormwater ponds in Edmonton to understand if there are characteristics about how we design stormwater ponds that do better at supporting aquatic insect biodiversity. We use a net like this. So this net is called a dip net. And this one, it's D-shaped, you can see. It's got a flat bottom and then a curved top. And it's a much sturdier material. And that's because the way that you use this is you put it into the water and then move it up and down against the rocks and the gravel and the sand that's on the bottom of the pond. And so it needs to be a tougher material. And then you collect it and then pour your catch into a bucket and sort through it and take out all of the insects. So that's another tool that we use for sampling insects. Finally, if we're interested in surveying insects that are sitting on vegetation, maybe things like spiders aren't going to be flying around landing on flowers, they're not going to be in the water. So to sample insects that are on vegetation, we use what's called a beat sheet. So this is a beat sheet. You can see it's just a sheet with corners in it, and I've made this PVC pipe frame So I would hold this with one hand, or I would get a helper to hold it, and I would hold it under a tree branch. So you can imagine there's a tree branch here, and then I would take this other PVC pipe and whack the tree branch. And the purpose here is not to hurt the tree. Do it gently enough that you don't hurt the plant, but what you do is you jar that plant very suddenly and repeatedly. And that causes insects that are holding onto the plant to rain down onto this sheet. And it's white so that we can very easily see the insects against the fabric. And then you've got a lot of insects running around here, and you've got to quickly try to collect them all before they escape. So you can either just, I'm just going to put this here for a second, can either just use your vial and try to scoop them up, but that doesn't tend to work very well. So what we tend to use is something like this. Both of these are called pooters. So a pooter, another word for it is an aspirator. It's like a vacuum that you create with your mouth. So this is the old-fashioned version that you suck in this tube, and that creates a vacuum in here, and then you put that against a small insect, and that sucks the insect into the collecting jar. However, I find that this old model doesn't actually work very well. I prefer this one where it's more you're directly sucking the insect. However, an important thing here is a filter, because otherwise you suck the insect into your mouth. So you suck up the insect, and when it's this big, you can suck up a lot of insects at once really fast, and spiders, and then cover this part with your thumb, open your vial, and then blow them into the vial. Whoops. So that's how, and then pop the lid on the vial. I do this better when I'm not trying to do it as a demonstration in my lab. And also, it can take two people. I often have somebody hold the sheet and somebody operating the pooter. So that's how we collect insects that are in vegetation. We would go along and we'd beat lots of vegetation along a whole transect and then collect all of the insects. Those are active sampling methods. Another way that we sample insects is with passive trapping methods. And for pollinators, this is one of the methods called pan traps or bowl traps. So these are just bowls, yellow, blue, and white, that we would fill almost completely with water and put a drop of dish soap in it. And then we would lay out these bowls in a field or wherever you want to sample pollinators and just leave them and come back about 48 hours later, and bees and flies and butterflies and other flower visiting insects are attracted to these colors. These are the colors that have shown to catch the most flower visiting insects when you make a bowl of this color and fill it with water. The reason why we put the drop of soap in is to break the surface tension, so that if an insect flies up attracted to this color, it hits the water and then it falls in, and unfortunately it drowns. But then we are able to collect the insect and bring it back to the lab and identify it. So that's pan trapping. So then after we've collected all those insects, we bring them back to the lab and we have to take each insect out of the vial and identify it. This is a very challenging part, and for my students, they usually spend several months, maybe like 8 months, identifying all the insects they caught in a field season. And so the main piece of equipment we use for that is a microscope. And so using the microscope, we also use a taxonomic key. So that's a book of information for the particular kind of insect that we're trying to identify. This one is the key to the bumblebees of North America. And inside there are couplets that tell you what characters to look for. And then it says if it's got a orange abdomen, go to A. If it's got a black abdomen, go to B. And then you know how to proceed through the taxonomic key, and you eventually arrive at a name for your insect. Before we get to looking at them under the microscope, some kinds of insects we need to mount on pins. Oops, I'll show you this one first. So because some insects, when they're wet, like the ones that we caught in the pan traps, are in water, their hairs sort of collapse and you can't really see parts of their body that you need. You need them to be dry and fluffy and like they are in nature. So with bumblebees, you actually have to wash them with soap and water and blow dry them, and then put them on a pin in a box like this. And then to identify them, you can just take out a bumblebee and look at all sides of it under the microscope, and you can hold on to the pin so you don't actually break any part of the bee because you're holding on to it. So that's the kind of equipment that we use in the lab. Is insect pins, boxes, microscopes, and taxonomic keys. And then at the end of the day, we end up with a long list of all the species that we found in that particular field border type, for example, or in that stormwater pond. So that's how we do our research in this lab.